Welcome to another Aspirational Neuroscience Journal Club. My name is Randal Koene. Today, I'll be presenting a paper by Ali Bahramisharif, Ole Jensen, Joshua Jacobs, and John Lisman. The title of the paper is Serial Representation of Items During Working Memory Maintenance at Letter-Selective Cortical Sites. Before we dig into the material, I'd like to introduce the authors. Dr. Bahramisharif, first author on the paper, holds a PhD in machine learning with work in BCI and is the CEO and co-founder of the company Machine to Learn in Amsterdam. Professor Ole Jensen is now running a lab at the University of Birmingham, where he's applying multiple investigative techniques to brain mechanisms that rely on oscillatory dynamics. His work is particularly significant to me because I was pulled into the study of multi-regional and multi-temporal brain systems through his early work when he published a series of papers with his mentor, John Lisman. The set of hypotheses and corresponding models expressed in those papers became the touchstone for my own PhD dissertation. I've listed the series of papers here. These here. There are many more modern ones, of course, but these here are still the foundational ones, I believe. In part of this presentation, I will occasionally borrow figures from my own papers to fill in some of the model context for the study conducted in this paper. Professor Joshua Jacobs runs a lab at Columbia University and specializes in direct recording in human patient volunteers. Work with patients is a unique opportunity to verify and validate hypotheses previously tested only in non-human mammals, and to conduct studies where the cooperation or response of a human participant is essential. And finally, there is the late, great Professor John Lisman, who passed away in 2017, but left an enormous legacy to neuroscience. To quote a small portion of the obituary published in Nature Neuroscience, his initial insight was that the secret of memory might be a molecular switch within individual synapses. He proposed an ingenious model in which individual molecules phosphorylate each other, forming a positive feedback system whereby a group of molecules can sustain a bit of information, even if individual proteins degrade over time. Soon after, calcium calmodulin-dependent protein kinase 2, CAMK2, was discovered. John then proposed a model for how synaptic plasticity could rely on CAMK2 for the maintenance of memories. These models heralded a lifelong journey to understanding the molecular basis of long-term information storage at synapses. Now, despite his strong theoretical disposition, John deeply cared about getting the answer right, and he wasn't afraid when experimental data, including the data from his own lab, proved his initial models were incorrect. Rather, such setbacks spurred him to update the model, proposing additional structural roles for CAMK2 as a master controller of a synaptic switch complex. This led him to devise important memory erasure experiments, published as his capstone paper shortly before his death. This last work, performed by a coterie of undergraduates in his lab, added critical evidence showing that CAMK2 is required for the storage of memories supporting a hippocampus-dependent behavior. Now, more to the point for this paper, another favorite topic of John's was brain oscillations. He noticed that the amplitude of fast gamma oscillations in a rodent hippocampus is modulated by slower theta oscillations. This suggested to him that each theta cycle could serve as a temporal frame for 4 to 8 gamma cycles. In an extraordinary intellectual jump, John connected these observations to working memory processing in humans. Psychology experiments had shown that humans have a limited working memory capacity of 7 plus or minus 2 items when searching for an item that is stored in memory, and it's serially scanned. John, together with Marco Idiart, proposed a model based on nested theta gamma oscillations that quantitatively explained these phenomena, with individual gamma cycles each representing an item in a defined order within each cycle of theta oscillation. Remarkably, this model has received much experimental support, from studies of the firing of hippocampal neurons in rodents to EEG recordings in humans during working memory tasks. In 1995, Marco Idiart and John Lisman proposed that multi-item working memory was supported by a mechanism in which individual memory items are repeatedly activated in sequence. All right, so we just mentioned the limited capacity of working memory, which was pegged at 7 plus or minus 2. In 2001, Randal Koene published summarized data that showed a typical limit closer to 4 plus or minus 1, making the 7 items more of a maximum for non-complex items. In addition to that, let's have a quick review of the essential capabilities that working memory must supply, given what we know about human memory acquisition. It must be able to remember multiple items concurrently, like this telephone number, for example, where you see that it's not a coincidence that the last digits are 7 digits. It must receive items to remember together at an arbitrary interval. So basically, if someone is telling you this number of 7, this 7 number telephone number, or 10 numbers in total, but 7 numbers in the last part, it shouldn't matter whether there is half a second between each digit, 1 second between a few of the digits, 1 second between one and another, and then half a second between the next two, and so forth. That shouldn't matter. In other words, there has to be a form of time compression. It should be able to catch and encode these items instantly. Now, maybe we're not always that great at remembering telephone numbers, but we can remember many types of episodic information instantly. We don't need to go through it several times to be able to remember that the apple fell from the tree. It should be able to sustain items in working memory, at least until LTP can take effect. LTP at synapses typically does not take effect after a single presentation of pre- and post-synaptic activity. It must be able to sustain items in temporal order. It must be able to sustain items in the temporal proximity that permits STDP. So if you are somehow maintaining items, you need to be able to reactivate them in a way where their activity will again be temporally proximate enough through time compression to make it possible to create some kind of an association between the patterns of the one item and the patterns of the other. And then this is not strictly a working memory thing, but the type of items, whatever an item is it's variable. It can be a complex item. It can be a simple item. There's this notion of item chunking that happens somewhere. I'm mentioning it here because it's closely related, but it's not necessarily something that's happening in working memory. Okay, let's compare here. Let's contrast this with long-term memory. In long-term memory, we know that long-term memory has a very large capacity, and the preponderance of evidence suggests that synaptic changes underlie long-term memory encoding and maintenance. By contrast, working memory has a very limited capacity. Here, the preponderance of evidence points to encoding and maintenance by ongoing neural activity, and that neural activity can maintain different concurrently held items and can do so in the same general network or subregion. Interestingly, retrieval time for an item held in working memory is correlated in linear fashion with the position of that item in a list. This is the result of the famous Sternberg task. In 2013, to review the collected empirical data relating to the distinctions between LTM and working memory and their dynamical model, John Lisman and Ole Jensen published a review paper in Neuron called the Theta-Gamma Neural Code. Let me list a bit of the comparison from that article. So in long-term memory, let's talk about gamma first. Studies of long-term memory have focused on the hippocampus. It was found that gamma power and spike gamma coherence in the monkey hippocampus were higher during successful encoding. Similar correlations have also been found in humans, both in hippocampus and cortical regions. Now for theta. In rats, hippocampal theta increases during locomotion or attention and is necessary for memory function. In humans, the theta power preceding the stimulus predicted subsequent memory. And successful memory formation was shown to be predicted by how well spike timing was phase coupled to theta oscillations. Recent work suggests that in humans, slow wave theta is predictive of correct recall and corresponds functionally to the 7 Hz theta of rats. And the slow wave theta in humans is a little bit slower at 3 to 4 Hz. Theta gamma coupling. Theta gamma coupling has been shown to be functionally important for long-term memory processes. And in one study, rats learned to associate contexts with the location of subsequent food reward. As learning progressed, there was an increase in cross-frequency coupling. Moreover, the strength of coupling predicted the probability of correct choice. These and related results suggest that theta gamma coupling in the rat hippocampus enables the recall of stored information. A recent study on humans points to a similar conclusion. Long-term memory encoding was associated with increased cross-frequency coupling between frontal theta and posterior gamma oscillations. Now, contrasting this, in working memory. Now, first I should say that in a typical working memory experiment, subjects are presented with a list of several items. This is followed by a delay period, usually less than 10 seconds, during which no information is presented. Subjects are then shown a test item and must make a response based on the properties of the information stored in their working memory. So for gamma, in humans, MEG studies and intracranial recordings have reported an increase in gamma power during the delay period of working memory tasks. Importantly, this increase varies with the number of items being held in memory. An increase in spike field coherence during the delay period has also been observed. Now for theta, several studies in humans have reported sustained theta band cortical activity during the delay period, pointing to a role for theta in working memory. Experiments in monkeys have shown that persistent firing during a working memory task is phase-locked to low-frequency theta delta oscillations in the local field potential, both in extrastriate cortex and in prefrontal cortex. Now for theta gamma coupling, theta gamma coupling during working memory has been demonstrated in humans, both in cortex and in the hippocampus. Gamma power in the hippocampus is modulated by the phase of theta oscillations during working memory retention, and the strength of this cross-frequency coupling predicts individual working memory performance. Importantly, the particular cortical regions demonstrating cross-frequency coupling depend on the nature of the information being held in working memory. The spatial distribution of gamma band power in cortex can be used to predict whether working memory is maintaining an indoor or outdoor scene, and the gamma activity with this predictive capability is phase-locked to the theta activity. In another study, gamma power at certain sites, primarily in occipital cortex, was shown to depend on the particular letter being viewed, and gamma. Study from 2009 by Jacobs and Maikana is closely related to the work in this paper. So all of this leads to the principal hypothesis behind what I'm just going to call the LIJ model, the Lisman-Idiart-Jensen model, namely the theta gamma code. Here we have that. The items in sequence, such as a sequence of letters that you see here, QDR, used in this task, are represented by spatial patterns of neural activity taking place within the interval of a gamma cycle. The activity corresponding to different items in a list takes place in consecutive gamma cycles. And the sequence of patterns of activity within gamma cycles is repeated on successive theta cycles. There are a number of important mechanistic requirements for the hypothesis, which were first presented in the form of the LIJ model and its support in the literature before 1995. An important requirement is the ability of the spiking of individual neuron to reactivate without reliance on pattern-specific synaptic input. Once input causes neural firing, that firing will need to reappear at approximately the alpha-theta interval. Now, a strong candidate mechanism for this is intrinsic after depolarization, the inverse of the well-known after hyperpolarization that suppresses membrane potential after a spike. ADP has been observed in types of pyramidal neurons in cortex and hippocampus. Published studies by Yue and Yari in 2004 and others describe how Kv7, KCNQ, M channels, a slowly activating, non-inactivating potassium ion conductance, modulates ADP in hippocampal pyramidal cell. Septal input at alpha-theta frequency would act to synchronize cycles of ADP, forming the slow clock of the system. As a side note, the difference between the fast 60 to 100 Hz gamma rhythm subtype that's observed to be caused by entorhinal input to hippocampus versus a slow 30 to 50 Hz gamma subtype driven by CA3 activity may support differential encoding of autoassociative patterns input into episodic memory pattern on the CA3 recurrent fibers. Septal input at alpha-theta frequency through GABA B receptors in CA3, which Does et al. And others have shown reduce both GABA and glutamate release at the target neurons, potentially further regulates the timing of new input versus episodic reactivation, thereby providing a temporal multiplexing of network function into encoding and retrieval supporting modes. Functional differentiation is further supported by the prominence of fast NMDA channels in temporal regions and the additional presence of slow NMDA channels on recurrent fiber synapses in CA3, facilitating nonlinear dendritic integration over consecutive inputs as tested by 2-photon imaging and glutamate uncaging by Makara and McGee in 2013. Here's how the LIJ buffer model comes together then. In the presence of acetylcholine, pyramidal neurons exhibit after depolarization. That after depolarization is regulated at rhythmic alpha-theta frequency. When input appears during the phase of theta, at which activation through input fibers is promoted, shown here first in the membrane potential trace of model neuron A and then that of model neuron B, the input is reactivated on subsequent theta cycles. As the second pattern activity appears, it is also reactivated, and its phase of reactivation precesses until it can go no further due to the suppression of activity by recurrent GABA inhibition that is caused by the network of recurrent fibers from interneurons triggered by the previous pattern of spikes. The two patterns now reactivate in consecutive slots, phase lock to alpha-theta. Shown on the left here, in a region such as CA3, repeated reactivation of patterns in the same order within a short time interval within the window of spike timing dependent potentiation can lead to LTP on synapses. So you see here this window between these two spikes. So it can lead to LTP on synapses between the neurons of temporally adjacent patterns, thereby encoding an episode of patterns of activity while also producing a time compression because of relative insensitivity to the delay between successive inputs to the system. Also shown is how the addition of further input eventually leads to the drop-off of the first pattern, resulting in a first-in, first-out type of short-term buffer of limited capacity, which is characteristic of working memory. And on the right, we see this zoomed out to show the arrival, maintenance, and drop-off of patterns A, B, C, D, E, and F. Finally, just for completeness, comparing the model behavior in the absence of noise on the left and in the presence of strong subthreshold noise on the right. Even in the presence of noise, and with a gradual reduction of the number of neurons representing each pattern, the general properties of a working memory can be sustained as long as a sufficient pool of pyramidal neurons is involved with each pattern. The series of papers from 1996 went on to demonstrate how the system of networks in entorhinal cortex and hippocampus could support immediate encoding and working memory, as well as early autoassociative pattern encoding in networks that feed into the hippocampus, shown here on the bottom, where then the recurrent fibers in CA3 support the first stage of episodic memory encoding. As demonstrated in numerous publications building on the work of Lisman and collaborators, this allows the development of a large variety of models that make verifiable predictions about observable activity during working memory-dependent behavior. One fairly simple example with place fields and phase precession is shown here. There are many others, of course, particularly where in vivo recording and behaving rodents can be compared directly with model output. In 2013, Lisman and Jensen identified and summarized studies that may further clarify the level of experimental evidence for the crucial hypotheses of nested oscillations and cross-frequency coupling, or CFC. Here in Figure 1, the neural code organized by theta and gamma oscillations. In A, we see simultaneous extracellular on the top and intracellular bottom recordings from the hippocampus. Intracellular gamma is due to IPSPs, the amplitude of which is modulated by the phase of theta. This is work by Pentonen et al. In 1998. In B, we see a schematic of the theta gamma code, which we've already discussed. The ovals at the top represent states of the same network during 2 gamma cycles. The active cells are black and constitute the ensemble that codes for a particular item, and different ensembles are active in different gamma cycles. In Figure 2, we see in A, phase precession. As the rat runs through a place field, a process that takes several seconds, theta phase becomes progressively earlier, closer to 0 in the phase on successive theta cycles for that particular cell, right, for a cell that you're recording. This is from Mehta et al. In 2002. The phase precession can be understood as queued time-compressed readout of the sequences of places ahead of the animal. Such sweeps provide information to other brain regions of what is to come. According to one hypothesis, the successive activation of cells during a sweep results from simple chaining of excitation between the ensembles. There is now little doubt that multi-item information is formatted by a theta gamma code in the hippocampus. Different spatial locations are represented at different theta phases, and firing is clustered into discrete periods by the gamma rhythm. These oscillations and their interactions are altered during both long-term and working memory processes, as would be expected if different neural operations make different use of these oscillations. All right, now with the foundations of the model and the studies that inspired it, here now are the new results provided in today's paper. The experiment is a direct test in human volunteers of 2 central hypotheses of the model. That elevated gamma power in specific cortical sites is involved with viewing specific items in the experiment, and that the alpha theta phase of that elevated gamma is a reflection of the list position of an item. Ultimately, the manner in which working memory is implemented and the maintenance of episodes is expressed, in other words, the hypothesis that it's done through time multiplexing, will affect how that memory is represented once consolidated in LTM through synaptic changes. So the data were collected in 15 epileptic patient volunteers who were implanted with ECOG grids during the mapping phase of their surgery for epilepsy at hospitals of the University of Pennsylvania and Thomas Jefferson University. All of the included data were from non-seizure periods. Data from brain regions identified as involved in seizure initiation and sites that had residual epileptiform activity were discarded. On average, data from 67 plus or minus 12 trials were used, and a sliding window FFT was used for time frequency analysis. The sites that were identified on the basis of stimulus-induced gamma power variation were in the temporal lobes and occipital lobes. And this is what the intracranial electrocorticographic experiment looked like. The patients were they were told to fixate on this cross, and then a number of letters were presented. Each letter was presented for 700 milliseconds, followed by a pause, a delay of 275 to 350 milliseconds. Then the next letter was shown for 700 milliseconds, again, a variable pause between that and the next letter. And then there was a maintenance period in which no letters were shown of 2000 milliseconds or 2 seconds. And then finally, there was a probe letter that was presented, and the patients were asked to respond. Now, there were 15 subjects, as we already mentioned, at a total of 13,015 sites, 1,315 sites. And the response time was on average 2011 plus or minus 412 milliseconds, with a 94.21% plus or minus 1.28% hit rate. So they were actually very good at responding correctly. Of all the sites present, letter-selective electrodes were identified by identifying those electrodes, those sites, where there was increased gamma power at 78 to 100 hertz for a specific letter. And then a mutual information metric was used by carrying out a permutation test to identify which letter that particular site was tuned for and which was its opposing partner. So for each of the sites, there was a tuned letter. There were tuned letters and untuned letters. For each site, what they did is they went through the various permutations and they found 2 letters with the greatest measured distance according to that metric. And in that pair, they would label the tuned letter as a tuned letter, the one with the high gamma, and then the untuned letter would be the one with the low gamma power. And for this, they then found these letter-selective sites in the occipital and temporal lobes, of which there were then 14 in 9 subjects. One of the subjects had 3 such sites, and 3 of the subjects had 2, and most had just 1. What we're seeing here is a time frequency analysis of the 14 sites. The theta, alpha, and gamma power at letter-selective sites are differentially affected by letter presentation. The phase amplitude coupling is shown here. Note that although gamma power is low during maintenance, this section here, it is not absent. So keep in mind that this is averaged over all letter-selective sites and that this is for letters at different positions in the sequence. In other words, if gamma phase is specific to position, it will be washed out in maintenance. And here on the non-selective sites, non-letter selective sites, we see that there are none of these obvious sites of periods of phases of elevated gamma power. Following figure, figure 3, shows CFC during maintenance. The CFC here was calculated by estimating the phase amplitude coupling for the data during the retention interval. What we see is that the CFC did not increase during maintenance, but be coupled to theta alpha phase. The figure shows strong 90 hertz around here, gamma oscillation power, and a specific phase of around the 8 hertz alpha theta. And very similar results were also shown in the recordings of local field potential in behaving rodents, and have been for years, for which there's a rich literature. These are just the top two parts of figure 4, just A and B. The C will be on the next slide. This is showing the properties of phase amplitude coupling during maintenance. In A, we see the average over 14 sites. The phase amplitude coupling, as you see here, is greater at the letter selective than at the non-letter selective sites. In the right panel, the differences here were calculated for each subject. The average difference is large, showing that phase amplitude coupling is much larger at letter selective sites than at the non-letter specific sites. In B, we see here the comparison between the tuned letter and the untuned letter. We see that the phase amplitude coupling is somewhat larger for the tuned versus the untuned letter, but it's really not a significant difference. And this is really panel C of the same figure. In this, again, we're carrying out this comparison. There's a significant difference here, doing a two-tailed t-test of gamma power between letter selective sites and non-letter selective sites during the maintenance period. There's no significant difference between theta and gamma power over letter selective sites when compared to the tuned letter or untuned letter during the maintenance period. And here the same for gamma. So we have this both for gamma and alpha. Overall, this shows that there's no significant difference in either the theta alpha or gamma power for tuned versus untuned letter selective sites, but a strong significant difference between letter sites and non-letter sites. The involvement in the task was clearly differentiated, and both oscillations are consistently involved. And I should mention that the authors were careful to ensure that the results were not due to phase locking effects of non-sinusoidal rhythms or due to performance, like reaction time differences between the subjects. And here we put it together to see what this means for the individual, for the position and the phase to position of an item in the list. So the preferred phase of the activation for tuned letters depended on the stimulus serial position in the list. The broadband gamma power is indicative, is higher at an earlier theta alpha phase for items that were presented in P1 than for P2, and at an earlier phase for P2 than for P3. So we have that the broadband gamma power is elevated at letter selective sites when its tuned letter is presented. Here we have it for P1, the red, then for P2, green, then for P3, the blue, at different phases of theta. And we see that the phase of theta alpha at which max gamma power appears varies with temporal position in the list. This is strong support for the serial organization of working memory as first proposed by Sternberg and modeled by the JLI or LIJ as it then became model, namely for this temporal multiplexing. The authors themselves make some future suggestions for further work, some suggestions for future work. They suggest that there should be more direct investigation of effects discovered by Kavanaugh about the inverse relationship of item complexity and gamma period or number of items. This is work in which it was shown that as the complexity of an item goes up, the gamma period becomes larger. It's holding a more complex pattern, and there's room for fewer items. At least this is how the model would interpret. There should be manipulations of memory load and memory content in similar ECOG experiments. Now, the LIJ model doesn't stand at odds with or apart necessarily from other models of memory. Together with models of synaptic encoding, particularly using STDP, this forms a proposal for dual memory mechanisms. In the first temporal phase of this dual system, momentary maintenance of working memory in cortical systems is translated into episodic synaptic changes in hippocampus. In the second phase, episodic associations are then entrained in cortical networks themselves, becoming independent of hippocampus after consolidation. And just to show where that fits in, these two different phases in sort of the whole path that memory takes, a peek at just a sort of a time span map of memory, where we could say in the first sort of 0.1 to 1 seconds, you have this moment where a stimulus is captured, and where it is coherent activity from perceptual processing that is giving you that stimulus. Then you have the second stage, which is where this working memory or short-term buffering is happening. You can hold something for a few seconds. It depends on the reactivation. Oscillations of phase coherent activations are keeping this alive. It's not necessarily specific to patterns of synaptic training or anything like that. Then you have what I guess I use this term, but others don't necessarily. I call intermediate term memory, which can last for hours, sometimes longer than that, and where you have LTP on synapses, and where episodic memories are still being maintained through hippocampal reactivation and being retrieved through hippocampal reactivation. Then you've got weeks within which there is further reactivation and consolidation during periods of slow wave sleep and other quiet waking moments in that system. And then ultimately over years, you get the reactivation of patterns, interleaved reactivation, presentation of new information, and the growth of associations between different patterns that are throughout the cortex and not dependent on hippocampus, as studies have shown. Thank you very much. And now it's time for questions.